Marcia and myself started preparing this conference, we asked ourselves what might be most interesting for you as an audience to hear. And so we started with that old saying from Stephen Jay Gould. Probably you have read some books of Stephen Jay Gould, one of the most eminent natural scientists of the 20th century. This is this idea of why are we here where we are and not at a different place? Why has our species survived and not other species? So this idea of getting one track on an LP or an old tape versus another track, which leads to the question, yes, why were you on one side of the ramification of the tree and not on the other side? And the title of our talk, Predicting the Past, basically tells about this paradox. We know that we all have the same roots, the same trunk, but we don't really understand why we are on one side of the ramification and not on the other side. And this is where we set in. So the first part of our talk will be like an introduction on what do we know about Neanderthals, humans, Denisovans, and other hominin species. The second part will be more a philosophical part. What kind of modeling systems do people use today in science? The third part is then the lab perspective on our own agent-based model of human dispersals. And the last part is an outlook, where to go from here. So first, what do we know about Neanderthals and ourselves? It was in 1857 when an amateur paleontologist proposed that Neanderthals are not just pathogenic humans, but that they are a real fossil species. And 140 years later, in 1997, we had the first publication by Matthias Krings and Svante Pääbo about Neanderthal mitochondrial DNA, basically showing there is no overlap between humans and Neanderthals. Later on, we had the modeling approaches, the first explicit spatial modelings of Neanderthals encountering humans and vice versa, which came to the conclusion that the mitochondrial evidence and then the nuclear evidence at that time would suggest there was no or very, very little interbreeding between the two species in Europe or Eurasia. So that led to the kind of the classical model, also supported by paleoanthropology and archaeology, that humans are an African species, Neanderthals an Eurasian species. There was a region of overlap in the Near East, but otherwise they were kind of a classical example of allopatric speciation, species diverging from a common ancestor and giving rise to very different morphologies, behaviors, lifestyles, etc., etc. But then, in 2010, we had the first more or less complete nuclear genome of Neanderthals, and the story changed quite dramatically. So this notion of interbreeding, admixture events, introgression events from those ancestral populations in modern human populations, expanding into the habitats of Neanderthals. And if you look at the picture that we have today, it's quite amazing. We have this significant contribution of Neanderthal alleles still living in our own genome. Some people say it's about 15 to 20% of the complete Neanderthal genome still present, dispersed over humanity outside Africa. And then we have, of course, the next step, even higher percentages of the more archaic Eurasian populations that are still just named the Denisovan populations. So that's the current picture, and probably the most amazing take-home message is that contrary, just like the opposite of the original thought, Neanderthals actually have human mitochondrial DNA and human Y chromosomes. Human in the sense of along the ancestral line of Homo sapiens. Whereas we have a lot of introgressed nuclear DNA from Neanderthals and Denisovans. So the story is complex and nobody is able to explain this outcome with our current knowledge. The archaeology also tells a very interesting parallel story. You have all kinds of population discontinuities among Neanderthals in a given cave. But you also have population discontinuities in terms of Denisovans, Neanderthals, humans, for example here in Denisova cave, which tells you a lot about population fluctuations, expansions, retractions, something that has been discussed by Mika in his first presentation. And then, of course, we have this recently published incursion of modern humans into Neanderthal territories. Probably incursion is not the best possible description of this phenomenon. Let's just call it population fluctuations caught at one specific location. So there are many open questions now regarding the patterns that we see in archaeology and paleogenetics and genomics. So which mechanisms can explain those patterns? And one question is especially interesting for us. We as paleoanthropologists are mostly focused on individual fossil finds, and we want to know how those individual properties of fossils, of populations, of species interact with the large-scale and long-term properties of climate fluctuations to get the pattern of population dynamics that we can derive or infer from the archaeological record. And of course, since we are here at a conference about climate and human evolution, the question is, what's the role of climate fluctuations? How can we address these questions? More data. This takes time. Excavating sites, that's normally a lifelong story, but we can kind of accelerate the process when we use models to simulate processes that would otherwise take centuries, millennia, and more. So this leads me to the second part of our talk. What kind of models do we actually have? Here you see just a very coarse classification of models into discrete versus continuous, something that has been discussed in the previous two presentations, static versus dynamic, deterministic versus stochastic. Now, what is a model? Basically, a model is just a representation, be it a physical representation as those that you see here, or a mental or a software representation of some concepts, plans, hypotheses, theories, etc., etc. So this implies abstraction and reduction. And now if we think about observations, experiments, and models, we could say, well, an experiment is somewhere between an observation and a model. The observation has just the natural system as a black box in front of it. That's the patterns that we see. And we start building hypotheses about those patterns. Then we do experiments. We constrain the natural system to produce some information. So we go from the black box to a gray box. And then in the third step, when we build a model, we actually assume we know the mechanisms. We know all the inner workings of the system. So the system becomes a white box, and we can now play around with states and see which state of the system leads to which outcome on the other side, which pattern. So basically we say we have a parameter space and some processes that result in an outcome space, and we want to know the connection between parameter space and outcome space. Is it more or less predictable and orderly? Is it chaotic, etc., etc.? Now there are two perspectives, and probably you're intuitively familiar with those perspectives. It's so to say the likelihood perspective and the probability perspective. You can ask, for example, given some parameter values, what are the outcome probabilities? given some properties of Neanderthals and humans, what's the probability that Neanderthals go extinct? What's the probability that humans go extinct? On the other hand, we can also ask, given some outcomes, we know that Neanderthals went extinct, what's the likelihood of certain parameter settings that would produce that outcome? So we have these two perspectives. And these two perspectives are basically everywhere where we speak about models. It's the likelihood perspective and the probability perspective. And now the question is, which model should we choose to model some specific question, to test some specific hypotheses in our computers? And I will show you this is mostly a matter of context and of taste, of scientific taste. There is no clear rule which model we should use. I will just give you some examples. As we have been co-working with astrophysicists while building our agent-based models of human dispersals, we became a little bit familiar with the jargon of astrophysics modeling. Here you see some of those keywords. It's basically particle-based, physics-based simulations. Climate models, I won't bring owls to Athens here. We are in the Climate Physics Center. But it's interesting to read, let's say, the meta text about those models. You see words like confidence, credibility, accepted physical principles. And I think every climate scientist knows how important it is to be credible, to have confidence in the models in a political perspective as well. Weather models. There are some very interesting developments in this field, kind of adding to the classical general circulation models some, as they call it, neural general circulation models. So you see, artificial intelligence, machine learning comes into the picture. Very interesting recent publication in Nature that deals with this topic. Infectious disease propagation. You have heard a lot about models and probably already forgotten during COVID times. Everybody projected their ideas about how the pandemic will evolve, and you see here in a publication from 22, sometimes the predictions were good, sometimes they were not so good because the parameterization of the model was probably very far from reality. The Human Brain Project, one of the most prestigious European projects that started in 2013 and completed in 2023. You see the amount of funding that was invested into the model, and the idea was to actually simulate the human brain that could think and behave. And suddenly people realized after about 1 or 2 years that it doesn't work. And the promises that have been made of simulating human thought simply cannot be met. Now, the outcome that we have from this project is interesting in itself. It's a 3D atlas of the human brain at the level of single neurons, not of the entire brain, but at least of some interesting regions. But has nothing to do with the original aim of the project. That's a classical, basic scientific endeavor to produce an atlas. But things actually worked out quite well for a much smaller brain that only has 140,000 neurons, the brain of Drosophila, the little tiny flies that swirl around when you open a good bottle of wine, for example. And here it was possible to actually simulate all the neurons and those 50 million synapses. And here you just see the 50 largest neurons of Drosophila, of a Drosophila brain. Just to show you, a neuron is not a single entity, it's a highly sophisticated parallel computing machine, each neuron in itself. So it's the network of neurons and this architecture which makes the model so interesting, and you can actually use this model now to simulate Drosophila behavior. Then we have heard a little bit about ecosystems models. There was this kind of pioneering publication more than 10 years ago about simulating all life on Earth. Now in the talk just before, to Shara's talk, we have heard simulating all the mammals on Earth, which is, as you have seen, a successful endeavor. AI models, I think I don't need to go into the details, but still the two basic publications, the Hopfield network, the Boltzmann machine that gained the Nobel Prize this year, set the stage how important this kind of modeling has become in recent times. But there is a problem. AI models are, again, black boxes. We don't really understand how they work. Now there is an extra research area, computational exploration of AI models to understand how they actually work. And then we have a second problem, that's the energy consumption of those modeling systems. Look at the numbers here, brain, 20 watts. That's a minor light bulb, and it can produce quite an amazing task. A supercomputing system, about 4 megawatts. Conclusion. Models are cultural objects. It really depends on the context, where you come from scientifically, what problem you're going to tackle, which model you then finally choose to proceed. And I put this slide up just to kind of encourage you to look a little bit around. What kind of modeling traditions could be used from other disciplines for your own discipline? And probably there is much more crosstalk possible between disciplines in creating models than we normally think. Now we come to our own model, modeling past human populations. Is it time to model all Pleistocene humans on Earth? Probably not yet, simply because we don't know that much about Pleistocene humans as we know about mammals in general. But just to give you a first example of how such models look like, these are now based on individuals that start dispersing all over the globe. This is Homo sapiens. So each single individual in this simulation is treated as an entity that is born, lives, mates, dies at some point. And on top of this are the climate data provided by Axel's group. And you can see how glaciers retreat, advance, how people move along water lines, major rivers. You see how the Sahara dries out and gets wet again. And finally, when the glaciers retreat into the Holocene expansion into the Americas. So that's just a straightforward model, no competition with local Eurasian archaic populations, just to show you how it works. Now, of course, our question is why and how did the archaic Eurasian populations disappear? Why did they die out? Is it climate-made? Is it human-made? Or is it both? Of course, we think it's both, but how do these two major factors work together? Now, what's extinction? Extinction is something quite complex. Extinction simply means there is no viable population left. But the reasons for extinction are normally multifactorial. So probably Neanderthals died out in the Iberian Peninsula because of resource competition. They probably died out here because of aridification, because of mammals overhunting, because of pathogen loads. We don't really know. We just know at the end Neanderthals disappeared. So the question that I mentioned before, I restated here, how do these short-term, small-scale processes of individual behavior interact with the long-term climate-driven and environmental change-driven processes to influence human dispersals, survival, and extinction? We call our model the ABM HPC model, agent-based modeling of human dispersals using high-performance computing. The model runs on a geodesic grid, which is equalized, so the cells have more or less the cells have the same size, and we use 3 different scenarios, modeling scenarios. One is a very simple sphere, only consisting of 162 nodes. The second one is a highly detailed sphere, but it only has continents without any specific properties. It's just a landmass that we model there. And the third one is the realistic Earth model, as we name it, which has time-evolving topography, climate, and vegetation. And this last model, that's the most complex one, you see how it is built. It has the time-evolving paleo environment, and it has the agents, the individual humans that we model one by one, and the interaction between the environment and the humans is basically via net primary production, local net primary production values in this approach. Do individuals matter in our model? Yes, of course they do. In the previous 2 presentations, you heard about the top-down approach using partial differential equations that basically think about individuals as microscopic, infinitesimally small particles, whereas we think about individuals as discrete entities that interact, that have their specific individual properties, such that the population dynamics emerges from the system and is not defined in the equation that we see here. Of course, at some point, the 2 systems should result in similar results. So our agent-based system should produce the outcomes of that equation if everything goes right in a simple environment. Now, let's go a little bit into the details of the models. How do we actually transfer those thoughts of population dynamics at the level of individuals? That's the classical Fisher-Kolmogorov equation that you see here, the diffusion term, the logistic growth term. And this is normally how people think about logistic growth. So when there is nobody around, you have the classical growth rate R, the maximum growth rate. And then with increasing population density, with increasing numbers of individuals, death rate increases and actual growth rate goes down. And when they reach the same value, you have no net growth of the population. That's at carrying capacity. But if you look at populations, at small populations, the situation is much more complex. You have, we don't really know how birth and death rates develop when population densities increase. But we know probably something about the turnover rate, which determines how many individuals die and are born at carrying capacity. And that's a key variable, because if that turnover rate is very small, as for example in great apes, the risk of extinction is much higher than in us humans, where the turnover rate is quite high. So that's something that is very important, but still it's the population view. So in our model, we take a very simple approach, which is more or less based on available data from hunter-gatherers. We assume that birth probabilities, birth rates are more or less constant, whereas survival probabilities, which means death rates, increase with increasing population density. So that's the individual perspective here, no longer the population perspective. Next question, how do humans move? Can we trust the diffusion term that we normally have here? Mika told us about weighing diffusion terms according to NPP, but there are more additional ways how we can think about diffusion. Is it really just random movement, or is it directed movements like in Levy walks, where you have long stretches of linear migrations followed by search patterns which look more like diffusion. There is an interesting publication that basically pretends that, or states that humans do follow the levy pattern of foraging. Now, we looked at those patterns in detail. Here you see the data from the Reichland study, 2014, and we mapped it to the actual terrain, something that the authors didn't do. So here's the terrain. And wow. So basically what we see is those levy-like Hadza foraging runs, they just reflect the environment properties. They reflect the terrain, which itself has this fractal structure of the riverbeds. So we transform that in our model that we say, similar to the approach of Mika, that our agents, our individuals, follow or have a tendency to follow the highest productivity cell in their neighborhood. So it's kind of a weighted stochastic function. Resource efficiency. Here we just took the data from Mika's 2018 study about the relation of net primary production to carrying capacity. You see, human carrying capacity increases with local net primary production, but there is a threshold. So there's a saturation effect, and Mika nicely showed that this is mostly related to infectious disease load. So you can't get out more of your environment. Also, it would actually produce more. So that's resource use efficiency. Now, these are the final equations that you can see here, very simple. Fertility from start to end, the relationship between net primary production and K max. And on the left side, you have the parameters that we model. So it's about 10 parameters in our system. And this is not too much. It's just what we need to model a realistic human. Now some computational considerations. If we just think about the order of magnitude of operations that we need to carry out one simulation experiment, it's about 10 to the 19th operations that we need. That's quite a bit of computing power that we would require to perform these tasks. And now we realize that it's not really efficient to parallelize agent-based models too much. You spend more time in parallelizing than you would spend time in just computing pleasantly parallel simulations. So you just let your parallelized simulation run in parallel. Let's say 10,000 different simulations, 10,000 different parameter settings, resulting in 10,000 outputs at the same time. So we start our simulations with two populations. We call them EAH, Eurasian archaic humans, and AMH, African modern humans. And we use this pleasantly parallel simulation approach. About 100,000 time steps per simulation run. Individuals grow, they mate, they give birth, they move, and they die. And on a global level, we just update the time-evolving paleo environment with the data provided by Axel's group. Now, here we show you just 4 examples. We seed a population in Africa. We don't really look that much at how they proceed in Africa, but then 4 different parameter settings result in completely different outcomes. So see, the one here at the bottom is, this seems to be a quite successful Homo sapiens population spreading over the different continents. The one on the top left is less successful. It gets stuck in the Nile Valley. It doesn't advance much. The one on the top right advances more slowly through Eurasia, And now we just wait until we reach about 20,000 years where these simulations stop and see what's the outcome. So you already see the outcome here, almost complete replacement of Eurasian populations by African populations. On the top left, no replacement, and the other two simulations give a kind of a more mosaic pattern of replacements. So this is now our classical approach. We just want to see what happens if we use some kind of parameter combinations for the two populations. What happens if we take, let's say, here the blue combinations, what's the outcome? Now, doing that is, again, very expensive. It's computationally extremely expensive. Even if we just have 3 values per parameter, low, middle, high, we cannot simulate this amount of data because we cannot handle it. We don't know how to handle it, and we don't have the time to simulate it. So what we do is basically we try sparse sampling both on the parameter space and on the outcome space. On the outcome space, it's more intuitive. We just sample some regions, about 100 regions, and a certain surrounding around the center. In the parameter space here, it's a little bit more complex. We use so-called Sobol sequences, which give us kind of a nice homogeneous distribution of sampling points in those 10 by 10 dimensional spaces of the parameter range. Here you see 9 outcomes of about 10,000. Now it would be nice to just look through those outcomes, but again we cannot do it. It's too much visual information. We cannot see a pattern. So what we use is 2 different techniques to actually gather and interpret the data. One is the classical approach. We use empirical orthogonal functions, for example, principal components analysis. The other is we use machine learning, so we basically train a system to understand our outcomes and see which parameters are really important for the outcome. Now, what's the outcome pattern that we see? In our featureless sphere, out of Africa and into Africa is about the same probability. As soon as we add continents, and especially if we add the realistic Earth model, allopatry is the predominant pattern. So separated populations in Eurasia and in Africa. And this is basically what we see in the archaeological record for a long period of time. But then you have interesting things like into Africa and out of Africa scenarios, but at much smaller proportions. And these are the 3 main out of Africa scenarios that we find in our simulation. That's the one that we know from reality, but then you see those with relatively stable local populations and a lot of overlap zones between those 2 different populations. Which parameters are important to get those results, to colonize a continent? It's independent of the scenario, of the model scenario. It's mostly birth rate and how efficiently you can use the resources. So how efficiently you reproduce and how you efficiently use the resources, and the interaction between many different parameters that we cannot really specify here. So that's a complex pattern and a relatively simple message that we can say at the end, at least. Now, when do people populate Eurasia? We can see all our simulated data basically coincide with windows of climatic opportunity, as we call them. As soon as you have a corridor, a green corridor, as soon as a window opens from Africa from sub-Saharan Africa into Northern Africa and then into the Near East and Europe, you find those out of Africa movements. And interestingly is that populations can be very similar. So populations, let's say, on the north side of the barrier and the south side of the barrier, but they can also be very different, which means the most important factor regulating out of Africa movements is climate. It's not necessarily the population themselves. Now some outlooks. Geography, climate and dispersals. Dispersals are relatively rare event, and we have to rely on the climate windows of opportunity. Entering Eurasia from Africa is difficult because you have to pass first a climate window, then a geographic bottleneck. But we have kind of an explanation why people didn't recolonize Africa. It would be even more difficult. First, your population would be decimated through the geographic bottleneck, and then you have to encounter the window of opportunity, which is normally closed. Partial replacements are the rule, extinctions are the exception in our model. And what we think is the reality, when we now go back to reality, is that we will probably find more evidence of fluctuating coexistence of Eurasian and African populations in Eurasia. Thanks to John David Jody Weissman and Ali Vadati, who implemented the actual agent-based modeling system. Thanks to the Swiss Platform for Advanced Scientific Computing, who funded our research. And of course, thanks to the spirit of ICCP. This picture was taken yesterday. You see Axel in the position of a kind of a voodoo priest preaching climate here. Thank you for your attention.